Good morning, everyone. Thank you for joining us here. Welcome to EPA's Computational Toxicology Communities of Practice. My name is Cameron Clark. I'm a student contractor in NCCT. Today we're going to be hearing from Steve Simmons, a research toxicologist in NCCT. And he is going to be presenting on mRNA transfection retrofits cell-based assays with xenobiotic metabolism. So if you have any issues with the webinar, please either speak up or type it in the chat box. But I'm going to go ahead and let Steve take it away. We're getting a little echo. So if you don't mind, your line.
The leader has muted your line. To unmute your line, press #6.
On the line, make sure you don't have it on speakerphone because it'll feedback. All right, well, I'm going to go ahead and let Steve take over. But again, for those of you who are on the line, please don't put your phone on speaker. You have feedback coming through. Make sure it's on. All right, do you mind giving me a try? Yeah, they should both be.
important problem is underestimation of chemical hazards in vitro if the parent compound is activated to a more toxic metabolite in vivo. And so here I give a sort of a classic example. If you've gone through a toxicology program or had a toxicology course, you learn this example, and that's of the polyaromatic hydrocarbon benzoate pyrene. In and of itself, in vitro assays, it's largely inert because you can see it doesn't really have any functional groups. But in vivo, it is metabolized by cytochrome P450, particularly CYP1A1, 1A2, and a couple of others, to form an epoxide, which is the metabolite you see on the right. And this epoxide form does form DNA adducts, which when misrepaired lead to mutations and causes cancer. And so benzoate pyrene is a known carcinogen in vivo, but one that's largely inactive in vitro because it's not metabolized. So we know not having metabolism in our test systems is a problem. And so when we look at what we're currently doing in terms of high-throughput toxicity testing, what we're talking about doing is we're talking about using cell-free and cell-based assays in high-density multi-well plate formats, so 96, 384, 1536 well plates. And we're using a lot of reporter genes, things that are either luminescent or they're not necessarily reporter genes, but analytes that are either fluorescent or luminescent, or change some sort of property of light that we easily measure in a plate reader format. Through our partnership with Tox21, we've introduced robotics into the system. So we're talking about pushing really fast, usually 10,000 to maybe even over 100,000 wells per day, and that's sort of our focus in terms of high-throughput toxicity testing. So our cell systems tend to be pretty simple. They tend to use cells that are immortal or tumor cells, immortalized or tumor cells, which do not have this metabolic capability, but are largely isogenic and give us a continuous supply of cells to test over and over and over again. If you go heavy into the world of metabolism, what you'll see is, the focus is very different. So the focus is on using biologically complex but biologically relevant cell systems that are complicated, a lot more complicated than what we use in high-throughput toxicity testing. They involve feeder layers of cells or matrigel sandwiches and things that aren't really necessarily amenable to high-throughput toxicity testing. A lot of the ways you measure metabolism in this world are low throughput, so using liquid chromatography coupled with tandem mass spectrometry, and you're looking, and a lot of times these are looking a little bit more mechanistically, right? So they're looking at how a particular parent is metabolized and what sort of metabolites, and getting ideas of measuring, the rates and things like that. And so it's a little bit more focused on one thing at a time, whereas in high-throughput toxicity testing, we're talking about testing thousands of chemicals at one time. And so even though we might use some of the same terms, there is a huge divide. And so how do we solve the problem? One way is to basically take what's already being used in the metabolism world and try to adapt it to high-throughput toxicity testing. So basically moving our high-throughput toxicity testing into something that's already metabolically competent. And so there are metabolically competent cell systems like primary human hepatocytes or HepaRG cells, but we have a lot of assays. So just in ToxCast alone, we have roughly 800 assays. Half of them are cell-based, and most of them are not in HepaRGs or primary human hepatocytes. In fact, virtually none of them are. And so we have to move a lot of assay technology to new cell types. Some of these are stable cells that have been published for over 25 years. It would be a really big lift to move all of our high-throughput toxicity testing infrastructure over to the metabolism side of this cliff. The other way we could try to solve this problem is to try to take the test systems we already use in high-throughput toxicity testing and try to just make them more metabolically competent. Since that seems like the more tractable approach, that's the approach we've taken. And we've got a couple of projects going on simultaneously. I'm only going to talk about one of them today, and it's an effort to retrofit our cell-based assays. But we do have, like I mentioned earlier, we do have both cell-free and cell-based assays. And so we do envision not only having two basic, if you want to bifurcate our assays into those two types of platforms, cell-based and cell-free, perhaps requiring two different solutions, but we also envision two sort of scenarios with metabolism in vivo. So one is where a metabolite is made in a tissue such as the liver, but then leaves the liver, finds its way into systemic circulation, and has an action, a biological action somewhere other than the liver, right? So what we call circulating metabolites. The other scenario we envision is where a compound is metabolized to a metabolite that's directly acting at the tissue in which it's formed. So you can imagine some sort of inhaled toxicant being transformed by cytochrome P450 in the lung and causing a lung tumor, right? So those are what we call direct acting metabolites. And so cognizant of both the fact that we have cell-free and cell-based platforms, and also that we have sort of two scenarios we'd like to model, we have employed two different strategies. One's called the extracellular strategy, where metabolism occurs outside the cell or in a cell-free system. I'm not talking about that strategy today. What I'm talking about is the one on the right, the intracellular strategy, where we render our cells metabolically competent and generate metabolites inside the cells and measure the biological effect directly on those cells. So how do you do that? Actually, there's a number of ways to do it, and it's not actually super new, this idea. As a matter of fact, I just showed you a paper from 17 years ago where somebody did it in Chinese hamster ovary cells with CYP3A4. But the general idea that most people use is to reintroduce xenobiotic metabolizing enzyme encoding genes back into cells that either have no or low expression. There's a number of ways you can do it: transfection, electroporation, viral vectors, etc. But typically these are DNA genes. They're driven by what's called a promoter, which is an on and off switch for that gene to turn on and off expression. And by expression, what I mean is transcription, this process of taking the instructions essentially encoded in DNA and turning it into RNA, which then goes through a series of processing steps into what's called a mRNA or messenger RNA, which then is basically translated by ribosomes into an amino acid chain, protein, and then a functional enzyme. So there's a number of reasons we wanted to do something a little bit different. And in the interest of time, because we lost a little bit of time at the beginning, I'm going to gloss over some of this. But the method that we're taking here is to transfect XME encoding, or that's xenobiotic metabolizing enzyme encoding mRNAs. It has a number of advantages, but the main one is that we bypass transcription. RNA transfections in the past have been kind of frowned upon because they've been toxic. They actually induce an interferon response in most cell types, so the cells think they're being invaded by a virus and essentially act like they're under some sort of toxic response already. So this is obviously something you normally wouldn't want to do if you're trying to measure toxicity in a cell-based assay. But there's been a number of advances in RNA synthesis over the past few years involving how the 5' caps are engineered, and then the use of what are called pseudobases, so substituting the typical uracil, thymidine, cytosine, and guanine that are used in RNA. There are pseudobases that you can substitute, and the nice thing is functionally it acts just like an RNA. As a matter of fact, you can actually enhance expression off these chemically synthesized RNAs. But because they look just different enough that they trick the cell into not thinking it's being invaded by a virus, so you don't get the interferon response or the cytotoxicity historically associated with RNA transfections. And the other nice thing about using RNA transfections is it allows the user to define the composition and ratio of input RNA. So if you want to mix and match RNAs, you can do that pretty easy just by mixing the RNAs you put into the pool. And so this is what we explored in this project, and so our method development focused on cytochrome P450 enzymes, which are in the realm of what we call phase 1 metabolism. So these are largely oxidation reactions. So I'm going to gloss over a lot of the work we did for optimization, but I just want to I'm trying to sum a bunch of work in one slide here just to say that whenever you do a system like this, or whenever you take on a project like this, there's a lot of things that have to be optimized for each cell type you might want to test. And so since this hadn't been done before, we were starting from scratch. But what are the things we need to optimize? Well, one is the transfection conditions, right? So for people who aren't familiar with transfections, most people who haven't done this before don't realize that RNA and DNA, or what we call nucleic acids, they're large molecules, much larger than the average protein. And they're charged due to their sugar phosphate backbone. They carry a negative charge. And so that's really sort of two strikes to readily crossing the cell membrane. Cell membranes don't typically like large molecules, and they typically don't like charged molecules. We can't really do much about the size of RNAs or DNAs for that matter, but what we can do something about the charge and about their sort of hydrophilic, hydrophobic nature. And one way you can do that is through what's known as cationic lipid transfection. So it's exactly what it sounds. These are positively charged lipids that essentially bind to RNA or DNA due to the chemical or due to the static attraction of the cationic lipids to the anionic nucleic acids, and essentially it coats it in something that's lipid. So whenever the cell membrane sees it, it readily takes up these capsules. So essentially they're like Trojan horses for RNA or DNA. And so with respect to transfections, there's a couple things you have to optimize. So one is how much the lipid to RNA ratio that you use, and that differs from cell type to cell type. And then the other thing is once you have that ratio optimized, how much of that you can actually deliver to cells to maximize, in this case, cytochrome P450 activity without inducing cytotoxicity is something that we hadn't empirically determined. So the first set of optimizations dealt with transfection efficiency, and then the next one was cofactor co-expression. And so the cofactor we focused on the most was something called P450 oxidoreductase, and it's required for electron transfer from NADPH to cytochrome P450s. And you can see the cycle there. There's at least two points at where P450 oxidoreductase donates an electron. And so without this, your enzyme cannot continue to cycle and metabolism doesn't occur. And so even though POR is ubiquitously expressed in virtually every tissue you ever tested, what's not known with any cell type you might try to do this in is whether or not the P450 oxidoreductase your cell makes is sufficient enough to support a lot of CYP activity that you're going to be putting into the cell with RNA transfection. So we found out that we needed to supplement. So we were in this project we were doing HEK293T cells, which is a human embryonic kidney cell line. They do make POR, but what we found was that setting aside 4% of our payload for P450, POR mRNA actually boosted CYP expression. So between the upshot of all of this is that the transfection optimizations and the cofactor co-expression optimizations doubled the amount of CYP expression we saw over just trying to shoot in just the CYP mRNA alone. So with our optimizations complete, the next thing we wanted to do was to look at the 10 most prevalent human cytochrome P450 enzymes in human liver, and we identified those through looking at a meta-analysis of over 700 subjects that was published a couple of years ago. One question we had internally was whether or not we should base our mRNA composition, if we were to pool a composition to make it look like human liver, should we base it off enzyme activity or mRNA levels, right? So testing human liver and seeing how much mRNA there are relative to one another is one question, and then measuring enzyme activity is a different question. But as you can see from the graph, this is all pulled from that paper, is you can see that it almost doesn't really matter which, that the rank order is roughly the same and the ratios are roughly the same. But for this project, we stuck with the mRNA composition levels. So these are the 10 cytochrome P450 enzymes we used in this study, and out to the right are the percent that particular CYP makes up of a human liver panel comprised of these 10 CYPs. So up until this point, we had done this work with luminogenic substrates for cytochrome P450s, which are a handy tool because they can be done in multi-well plates, and they can just be analyzed with a standard plate reader that lots of labs would have for measuring luciferase. But they're not widely accepted as being sort of definitive proof that your CYP enzymes are working as advertised. And so what we did was we tapped the brakes, and once we had our optimization completed and we felt like we had maximized the CYP activity in the cell model, we did a number of what are called benchmark substrate studies. So these are studies that are low throughput, and they use LC tandem mass spec to detect not only the parent compound and look for its depletion over time, but also look for the formation of a predicted metabolite over time. And because these are low throughput, we only did a handful of these that were sort of well characterized, but so I'm going to show you the 4. So on the right, we have CYP3A4, and on the y-axis is the concentration of its 6-hydroxy metabolite of testosterone formation over time. We took these studies out, or we collected samples over 18 hours. And so what you see is the top set, the green squares, represent what we would call the singlet. That is the transfection group that contained only CYP3A4. I mean, all of these groups have 4% P450 oxidoreductase, but the remainder of the payload is 100% CYP3A4, and you can see we get a fairly linear formation of the 6-hydroxytestosterone metabolite over 18 hours. And then the blue and red squares are two mixes. The red square is the liver mix, so it was what I showed you on the previous slides, all 10 CYPs pulled together. That liver mix is comprised of 27% of that liver mix is comprised of CYP3A4. The blue dot is a nearly identical mix. It has 27% CYP3A4, but it does not have any other CYPs. The remainder of the payload is made up with beta-galactosidase as a carrier mRNA. And then the black dots at the bottom have no CYP3A4. That payload is made up entirely of beta-galactosidase. So it's the same pattern for all of these. So on the right is CYP2E1, and we were looking at the formation of the 6-hydroxy metabolite of chlorzoxazone. Again, you see we get a fairly linear formation of that metabolite over 18 hours. We didn't test beyond 18 hours, so we don't know how long metabolism goes beyond 18 hours. Most of these benchmark studies are conducted from anywhere from about 15 to 30 minutes, typically, for hepatocytes. So we thought that we were doing more than due diligence going out 18 hours, and to be honest with you, we didn't expect to see activity sustained after 18 hours. So we haven't tested beyond 18 hours to see how persistent this activity actually is, but we know it's good through 18 hours. And the other thing is that most of our in vitro assays, those particularly cell-based ones, are 24 hours or less. So, one other point. So you can see that the red and blue dots on the right graph are a little bit lower than what you see for the graph for CYP3A4 on the left, and that's because the liver mix and the other mix with beta-gal, those two mixes only have, or they're comprised of, 2E1 only makes up 12% of that mix. So this is the next panel, and it's looking at CYP2C9 and the 4-hydroxy metabolite of diclofenac, and again we get pretty linear formation of that metabolite over time. And you can see the two dots in the middle where the mixes are comprised of 21% CYP2C9. And then on the left is 2D6, and we're looking at the formation of dextrorphan, which is the metabolite of dextromethorphan. Again, we get linear formation of the metabolite over time. And here you can see a really pronounced drop-off with the mixes, but it's because 2D6 only comprises 3% of the liver, and so there's not a lot of 2D6 in those mixes. So how do we compare to what we consider gold standard xenobiotic metabolizing competent cell models? And so what we did was we actually pulled data from the literature and from spec sheets from a couple of sort of widely used commercial sources for hepatocytes, and then for the liver, for HepRG, we went to the literature and found as many values as we could. And where people reported out rates of metabolite formation in picomoles of metabolite formed per minute per million cells. So everything here standardized to the same units. And so what you can see in the upper left graph, that's 2C9 metabolism diclofenac. You can see HepRGs in sandwich culture primary human hepatocytes don't do a very good job compared to suspension primary human hepatocytes. And suspension hepatocytes are essentially, that's the best you can get, right? And what you can see is that our HEC293 T cells, they're not liver cells like these other three models. They're kidney cells. They don't do this. This is not their primary job. But through ectopic expression of these enzymes using mRNA transfection, we don't reach the suspension hepatocyte levels of metabolism, but we definitely surpass the HepRG in sandwich culture. And for those of you who are on the phone who might be wondering, these are this was 2D HepRG cultures only, no 3D cultures. And then for on the right, upper right, is 2D6 metabolism of dextromethorphan, and it's the same sort of pattern. Our cells outperform HepRGs in sandwich culture hepatocytes, but not the suspension cells. Same for 2E1. The one area where we were really lacking was in the 3A4 metabolism of testosterone. And based on the pattern of the other three metabolites, I do not suspect that we really lack a lot of 3A4 metabolism in our cells. What I suspect is going on here, although we've not tested it, it's only a hypothesis at this point, is that you need a certain subclass of organic anion transporters to effectively get testosterone into cells, and our kidney cells don't express those to the best of our knowledge. And so what our poor performance in testosterone largely reflects is our inability to get testosterone into the cells, not necessarily metabolize it. A couple of things I want to point out here. One is the earliest time point that we measured was 1 hour, and the primary human hepatocyte and HepRG data were largely, again, 15 to 30 minutes when metabolism's at its zenith. So our competitor cell models have essentially a built-in advantage there. The biggest built-in advantage, though, that the liver cell models have against our cell models is that none of these 4 benchmark substrates, not in any of the studies that we looked at, nor the tech sheets from the commercial sources, they all used, we use DMSO as our solvent to solubilize these substrates. No one else does. Actually, no one in their right mind uses DMSO to solubilize their substrates for CYP studies because CYPs, it's just largely known, inhibit CYP activity. However, we wanted to basically do these LC-MS studies under conditions that we would be doing high-throughput screening. It didn't seem like it made a lot of sense to us to basically test our system under some sort of Pollyannish conditions just to see how well we stack up against primary human hepatocytes. What we really wanted to measure is how well we do under actual screening conditions, because our intention is to deploy this to high-throughput toxicity testing. So we tested it under those conditions. And so I'll touch on that a little bit later about how much of an impact screening, doing these studies in DMSO had, to the best of our knowledge. So with our optimization complete, we had reached a number of conclusions. So one is that we could retrofit a deficient cell model with robust cytochrome P450 activity. We knew that the cytochrome P450 activity was rapid, so we were reaching sustained levels of CYP activity 6 hours post-transfection. And we knew that we had activity that was fairly linear, at least over 18 hours. We knew that we were producing the predicted metabolites and at rates that exceeded 2D HEPA-RG models in sandwich culture hepatocytes, even when we handicapped ourselves by using high-throughput screening conditions. However, we never really thought going into this that getting cytochrome P450s to work in 96 or 384 well plates was going to be the hard part. Like I said, other people have done it. I showed you the study from 2000 where they overexpressed CYP3A4 in Chinese hamster ovary cells. The real problem is what happens when we couple this method with an existing cell-based assay, and do we observe CYP-dependent shifts in bioactivity? So for our sort of demonstration study, what we did is we stuck with HEK293T cells and we transfected them with one of the top 10 CYPs in the liver, the liver mix, which is the mixture of those 10 CYPs in the right ratio, or a beta-gal control. So we have 12 biogroups or 12 transfection groups. Those 12 transfection groups were tested with 56 compounds over 11 concentrations using a 36-hour exposure in triplicate, and then we measured cytotoxicity or cell viability using cell titer glow. I'm only going to show you results from a couple of the compounds, 4 actually. So the first one is aflatoxin B1, which is a known hepatocarcinogen. And what you can see with the black line and black dots is that in the beta-gal transfected cells, so these are cells that do not have any appreciable CYP activity, aflatoxin B1 is a borderline active, but only at the highest concentration. However, when we overexpress CYP3A4 in the green, CYP2J2 in the blue, or the liver mix, which has 27% CYP3A4 and 1% CYP2J2, we see a pronounced increase in cytotoxicity from aflatoxin B1 exposure. The second compound on the right is amodiaquin, and this is kind of interesting result. If you look, the 2 lines in the middle, the black line and the red line represent the liver mix and the beta-gal transfected cells, so the cells with no CYPs. And what you can see is that we don't really see a shift in potency, but we do see a shift in efficacy with CYP2C8 expression. It becomes much more cytotoxic at the higher concentrations. But interestingly, we did see a right shift in the mid to medium concentration range with CYP2J2, which was completely unexpected. And like I said, all of these compounds were tested with all 12 biogroups. These graphs have just been cleaned up to show you the significant ones. Another compound, amitriptyline, was bioactivated by CYP2C19 expression. And then the compound on the right, azobenzene, was bioactivated by CYP2E1 expression and the liver mix, which was 12% CYP2E1. The thing I'd point out, I don't know if I have it. Yeah, so amiodarone has only been tested in 64 assays out of the 800 or so, and it was only active in 2 of them. So it only has a 3.1% hit rate, so it's largely inactive in our in vitro assays. Amitriptyline has a little bit higher hit rate, 12.5, but it's interesting to note that most of the assays, I think half of those hits or half of those assays in which amitriptyline is active was in the ToxCast Adagene platform, which was done in a HepG2 cell line with what they call its proprietary enhanced metabolic capability. We don't know exactly what that is, but Adagene has told us that they have supercharged metabolic capabilities. So that explains half. So if half of the hits for amitriptyline were in something where metabolism wasn't just from the wild type cell. And then with azobenzene, it's been tested in virtually every assay that we've have in our inventory. It's only active in 14 of them, and half of those were the Adagene platform. So what did we learn? Sorry, I was looking at the time. We did not see a whole lot of detoxification with CYP expression, which is odd considering the role that metabolism plays in toxicokinetics. So I told you in the very first slide was that largely the whole reason we have metabolism is essentially to take things that are nonpolar and potentially toxic and to polarize them and make them less toxic so that we can excrete them and get rid of them. And so I was surprised. I would have, if I was to basically lay a wager before I started on this project or this particular experiment with the 56 test compounds, I would have wagered that we would have more detoxifications of cytotoxic chemicals than bioactivation of non or not as toxic chemicals. But we didn't see that. So it got us to asking the question, why did we not see that? So one possibility is, we just got the wrong test set of chemicals, right? I mean, I did stack the deck with cytotoxic chemicals because I wanted to see some of both, right? So we hedged our bet by having roughly half chemicals that we knew to be cytotoxic and roughly half chemicals that were either marginally cytotoxic or not cytotoxic at all. And it's only 56, so if you test, say, 5,000 or 10,000 chemicals, maybe you see some things that you miss. It's actually a sort of a small sample drawing big conclusions. That's one possibility. Another one is that because we've enhanced the phase 1 metabolism or the oxidation reactions, we've essentially overwhelmed the endogenous phase 2 metabolism of our test system. I don't know if that's true or not. I mean, I know that HEC293Ts express, they express glutathione transferases and UGTs, and they express we know for a fact they express some of the phase 2 metabolism. The question is, again, it gets back to like what we were discussing with P450 oxoreductase. Have we essentially, with our exogenous expression of cytochrome P450s, overwhelmed the cell's sort of endogenous levels to basically conjugate what we're making? So we have a lot of unconjugated oxidative metabolites running around the cell. That's another possibility. But the third one that we didn't really think about until we started analyzing the data was kinetics. And I don't want to bore everybody to death because whenever I learned this in graduate school, I thought it was extremely boring. But now it's actually turned out to be really, really important. And that is something called Michaelis-Menten kinetics. And so I'm going to make this real simple. Essentially, what we have on the left is a graph. The y-axis is the V, or velocity of the reaction. Just think of that as the reaction rate, how fast the reaction goes. And on the x-axis is the concentration of your substrate. So in the case of cytochrome P450 reactions for metabolism, we're talking about the cytochrome P450 enzyme's ability to attack a test chemical or parent compound and metabolize it, right? And what Michaelis-Menten kinetics say is that as we increase the concentration of our test compound, we can actually increase the velocity of the reaction up to a certain point. And so that certain point at which you can no longer increase the velocity of the reaction is what is referred to as the Vmax, and the concentration of substrate at which you are at half that theoretical maximum velocity is what's called the Km, right? And so the upshot of this is if our test compound concentration is well below the Km, right, so to the left of that vertical dash line, then the reaction is going to be very slow. So if we're 1 log unit below the Km, we're only operating at about 9% the maximum velocity. So the reaction is going very slow. On the other hand, if we're well above the Km, so if we're, then it increases the velocity towards the Vmax. So if we're at 10 times the Km, so 1 log unit to the right, then the reaction is occurring at 91% its theoretical maximum. And so a log unit in toxicology, or a log unit in vitro testing is not much. I mean, our test ranges normally span several log units. But this has real-world consequences as we think about retrofitting our assays and doing high-throughput toxicity screening. So I want to go back to the benchmark substrate study for a second. So you've already seen the graph on the left. That's the formation of 6-hydroxytestosterone by CYP3A4. What you did not see, what I didn't show you, was I didn't show you the parent depletion graph. We didn't actually measure the parent, and so what you're looking at is the depletion of testosterone over time in the same exact sample. And so while the formation of the metabolite looks nice and linear over 18 hours, we have basically 7 micromolar. The concentration of the metabolite at 18 hours is 7 micromolar, which essentially represents 2.2% of the parent compound. If you look on the right, we put in 320 micromolar of testosterone into our test system, and you can see that the 7 micromoles that we metabolized barely put a dent in the parent. So we're not clearing a lot of the parent, right? And on the right graph, I've actually added a red dotted line there at 100 micromolar. That's typically the top concentration that we would test at high-throughput screening conditions. Now, I said earlier that testosterone doesn't get into these cells very well, and so this probably isn't the best example, but it highlights something I think it's important, because if I showed you chlorzoxazone, the data looks exactly the same, and there's no reason to suspect that chlorzoxazone doesn't get into the cell. But the bottom line is if we're testing, so here we're testing at the KM or a little above the KM. Actually, we're at 3 times the KM here with testosterone, and we're still only clearing about 2% of the parent. And so you can imagine if testosterone were a toxic compound, and we're only clearing 2% of it over time, we're not going to see a huge right shift. We're not going to see a huge detoxification. On the other hand, there's a chemical like diclofenac, right? So here's diclofenac, 4-hydroxydiclofenac formation on the left by 2C9, and then depletion by 2C9 of diclofenac on the right. And here, we're also at 3 times the KM, so we were at about, I think, 28 micromolar for this study. And what you can see is that at 18 hours, we had metabolized 100% of the parent. And so these are just 2 chemicals, but they kind of span the opposite ends of the spectrum here of what we might expect to see. It should be pointed out that the Vmax for the 2C9 reaction with diclofenac is about 10 times as high, or 10 times higher than the Vmax for testosterone and CYP3A4. So even if we got great import of testosterone into CYP3A4, we wouldn't expect to see a lot of clearance of testosterone at over 18 hours just because the Vmax is just lower. And that's just the kinetics of the reaction. There's really nothing we can do about that. So bottom line is that since we don't know every enzyme, right? So if we were to test, say, 1,000 ToxCast chemicals times 10 different CYPs, that's 10,000 chemical by enzyme potential interactions, and we do not know the Km or the Vmax for any of those potential interactions. So we can never really know going into it a priori how much of a particular parent we should expect to clear. We can't change the Vmax or the Km. They are what they are. But one thing we could theoretically change, based on that graph I showed you from Michaelis-Menten kinetics, is we could increase the velocity of the reaction by increasing the test chemical concentration. However, as I mentioned earlier, since our libraries are solubilized in DMSO, that quickly becomes a sort of a diminishing return because the DMSO will inhibit our CYP activity. So in trying to increase the velocity of the reaction by increasing our test compound, we're also increasing the amount of DMSO in the system. And that would have a negative impact on the reaction. So just to show you, this is I looked. Most of the information I could find about DMSO inhibition of CYPs was done in cell-free systems with either recombinant enzymes, microsomes, or S9 fractions. And it dawned on me that I have everything in the lab I need to test this in a cell-based assay myself. I don't really have to go to the literature. I can just generate numbers doing the exact, basically under the exact same conditions we tested under, and so that's what I did. Again, we're looking at 293T cells, looking at CYP3A4 activity, and then what I did was basically just titrated in increasing amounts of DMSO and then measured the amount of CYP3A4 activity that we have normalized to no added DMSO. And what you can see is that the concentrations that we were testing at throughout the study, which was 0.5%, which is the typical high-throughput screening concentration of DMSO, we get 32% inhibition of CYP3A4. It's also known that not all CYPs are as sensitive to DMSO as others. CYP3A4 is probably in the middle of the pack as far as the 10 CYPs that we tested. A CYP like CYP2E1 is much more sensitive to DMSO than 3A4. But since this one's sort of in the middle of the road, I'm actually testing a panel of them right now. I just don't have the data ready yet. But if we can just use a 32% figure just for illustrative purposes, I wanted to show you. This is a graph you've already seen. This was the 2C9 metabolism diclofenac. So what is 32% inhibition? Because we did this study using 0.5% DMSO. that's the impact that having DMSO in our test system made on that study. So it doesn't completely close the gap between our cell system and suspension hepatocytes, but it definitely makes an impact. So what else did we learn? Well, I think it stands to reason that if we don't appreciably deplete a toxic parent compound, we're not going to see very many detoxifications. But it also assumes, right, that the metabolite that we're forming itself is not also toxic, and we won't know that usually either. And so I will point out again, stress something I said earlier, is that detoxifications are not a chief concern, and they're not our primary concern. Out of the two problems I said that not having metabolism presents, this is the lesser important of the two, but still I thought it was a little disconcerting that we didn't see more detoxifications. The more important problem was the detection of bioactive metabolites. That we did see, more so than I would have expected, even when using cytotoxicity as the endpoint. And I say that because we use cytotoxicity because it was quick, easy, it's easy for people to understand. But if I was, engineering a perfect study, cytotoxicity would not be the endpoint I would use to look for reactive metabolite formation, simply because cells have a lot of tools at their disposal to deal with reactive metabolites. Phase 2 metabolism, they have oxidative stress responses, there's all kinds of things they can do. So if you break a jar of pickles on aisle 7, they're going to call for a mop and a bucket, and if you look 18 hours later, it might have actually happened, but you're just going to miss it. And so what I would advocate for is the use of some of our pathway-based assays like endocrine and stress responses. I think they're just going to be more sensitive endpoints. I'll show you a slide on that here in a second. And the other thing that we learned was that pooling the mRNAs to mimic tissue metabolism like we did for the liver really dilutes the effectiveness over using an mRNA encoding a single enzyme, especially for the poorly expressed genes. So if you want to look at, if 2D6 or 2C19 or 2J2, which I showed you in some of those examples in the cytotox study, were, capable of bioactivating certain compounds, if you use this in a liver mix where you only have 3% 2D6, 3% 2C9, and 1% 2J2, you're going to miss those. But I will say that this transfection method does imbue deficient cell models with robust xenobiotic metabolism activity. It does let you control gene delivery in a way that lets you control expression in a way that typical gene delivery methods do not, so that titration of exactly 4% poor, if I showed you the graph, what you'd see is if we went above 4%, it actually hampered CYP activity. And so be able to tightly control the ratio of P450 oxidoreductase to cytochrome P450s is really important, and it's very hard to do with some other method other than what we're proposing here. Bypassing transcription RNA allows you to get to enzyme much quicker, and that's really important where time is a factor. So in 1536 well plates and 384 well plates, you get a lot of evaporation and edge effects with really long studies, and so not having to wait a day, like with DNA transfections, they normally recommend you wait 48 hours after transfection before you treat your cells, and then you have your exposure period. So you could be in a 384, 1536 well plate for 3 or 4 days, depending on the endpoint, and that just doesn't work. It's very cost effective, so everything, including the lipids, everything was less than $20 a plate, so it's 5 cents per well, and that was a pilot scale synthesis, so if we were to scale this up and have huge RNA synthesis, we'll probably get that cost down much lower. But the really most important thing is this method doesn't provide any real impositions on current cell-based assay protocols. We only did forward transfections for the manuscript, but in the original pilot studies, we did both forward and reverse transfections. They worked equally effective. And so your transfection mix can just be made and added to your cell slurry at the time you plate your cells in the plates. You don't really have to add any steps. Moving forward, right now we have a proposed partnership with our Tox 21 partners at the National Toxicology Program and at NCCT. It's under consideration by the leadership. And the idea is to compare this mRNA transfection method to a method they've already got up and going in Rockville with direct microsome addition. And so what we're proposing is to use a panel of about 6 to 8 assays to get a more comprehensive picture of what works well rather than trying to make a determination based on just 1 or 2 assays. And so what we have proposed is what I mentioned earlier, looking at cell stress pathway-based assays and endocrine assays. So we've already began adapting this to a second cell line. So they're called VM7 cells. They used to be called BG1 cells. But this is a cell model that's been used to study estrogen receptor transactivation. Basically the VM7s are an MCF7 variant, so this is a breast cancer cell line, so it bears nothing in resemblance to the 293T cells that you've seen up to this point. But we haven't even really done the optimization for mRNA transfection. We just basically went with the defaults that we had learned from the HEC293T studies, just to generate some quick data to see if this works. But the idea here is that methoxychlor is a chemical that has minimal ER agonist activity on its own, but it's demethylated by a certain subset of cytochrome P450s to a hydroxy metabolite, and then to another metabolite called HPTE, and both are much more potent and efficacious agonists of the estrogen receptor. And so when we transfected them with two of the CYPs that we suspected from the literature or known from the literature to activate methoxychlor to the ER-active metabolites, which were 2A6 and 2C9, you see that we do get not only a left shift, but we see an increase in efficacy of ER transactivation with methoxychlor compared to the black dots in line, which is the beta-gal transfected cells. We included CYP3A4 as well because CYP3A4 is known not to activate methoxychlor, so we thought it would be good to have another CYP in there that didn't do. If every CYP did this, then we would maybe think it's just due to CYP overexpression. But it was encouraging to see that CYP3A4 essentially was indistinguishable from beta-gal in this experiment. But this just goes to show you, at least for one chemical, we're actually doing a panel now of about 60 chemicals with different activities across different CYPs to see if this holds true for more than just methoxychlor, right? So if we can take a panel of chemicals that are known to be metabolized into ER-active metabolites, does this work, right? So this is just a pilot study for methoxychlor. So with that, I want to thank my collaborators, and I guess we've got about 5 minutes left. Sorry I didn't leave much time for questions.
No, you're good, and thank you, everybody, and especially Steve for bearing with our audio problems. But yeah, we do have 5 minutes, so if there are any questions or comments on the line, in the room, you're welcome to type them in the chat box. Please speak up.
Steve, I actually have never worked with these cells, so this is only just for my own personal understanding, but during your optimization process when you were transfecting these cells, it seems like you were concentrating on using a chemical process. Is there a reason why or why not you would use something like electroporation or something like that? I guess.
And if you don't mind repeating the question for folks on the line.
Right. So the question is why we focused on using a chemical method to introduce RNAs into HEC293T cells and not some other method like electroporation. And the answer is not going to be a satisfactory one. It's just that lipofectamine is something you can pipette in, doesn't really require any special equipment. And so I don't have any electroporation equipment in the laboratory. And so we started with this. It seemed to work. But it's not to say that electroporation might not be just as effective. We just haven't tested it. So.
Looks like we might have one coming in on the line here. We'll give them a couple minutes. So can we also highlight that CYP activities are limiting for detecting toxic metabolites, need to overwhelm the phase 2 enzymes in the cells? And it's from David G.
Hey, Dave. No, I wouldn't necessarily jump to that conclusion. That was why I gave all the caveats about using cytotoxicity, right? So you can imagine a scenario, though, where you're making reactive metabolites, and if given time, they're cleaned up. But if you're catching them in real time, you can measure things like, say, NRF2 activation, right? So I wouldn't necessarily say that, and we necessarily have to overwhelm every cellular defense to see them. One of the things we have talked about maybe doing, because some other people have done this, is they've looked at metabolism of carcinogens in like NRF2 deficient mice and things like that, and shown that if you do disable some of these mechanisms, you can build a model that's much more sensitive for detecting reactive metabolites. And I think we certainly could do that, right? So we can knock things out with CRISPR or use siRNA to diminish certain to basically knock down certain genes from being expressed so that we can essentially turn off a lot of the defense mechanisms. But at some point, you've really kind of got a Frankenstein model, right? So I mean, we're trying to come up with the simplest retrofit to imbue these models with metabolism. I do think that that might be a more interesting approach if we want to do something a little bit lower throughput and more mechanistic to basically get an idea of what exactly is going on biologically in terms of metabolism. One of the other questions, sort of, it's not what you asked, but sort of related, is that to me boggles the mind, is people have asked us, well, if you screen, like let's select through this cross-partner partnership, if we screen 10,000 chemicals across 8 assays, and in any one of those assays we find, say, 200 new hits that we didn't see before, are we going to go through and do LCMS to find out what the metabolites are for all 200 of those chemicals? And to me, that seems almost like a Herculean effort to find out every metabolite that we might possibly be forming, or and it assumes, I think, that there might just be 1 or 2 metabolites. There could be a constellation of metabolites that we're forming with any one SIP or pool of SIPs, and I just don't know. We might have to do it. We might have to do it with an assay or 2, with a handful of case studies to show that it can be done. But I just don't see where we would have the. I just don't see where we would have the bandwidth to basically do sort of a non-targeted study for so many samples.
No, he was just saying thank you.
Oh, and so within a lot of these cell series and have cell series in water culture, is there any look at looking at the liver, you have multiple types of cells, cells, and also within just the hepatocyte for the liver, you have batteries of cells that are more hydrogenated, there's more oxygen, there's less oxygen, so the surrounding is very different, so the metabolic constants might be different. So is there a way, like, comparing that to what we see.
Right. So the question was, basically there's sort of a heterogeneity of tissue, of cell types in, in liver or in any tissue for that matter, and there's sort of geographical differences with respect to, oxygenation levels and metabolic competence even within the same tissue, sort of creating these zones. And the question was, is there any way we can look for that? And that gets back to that slide I showed earlier with the big sort of canyon with high-throughput toxicity testing on one side and metabolism on the other. I think a lot of the extant tools that we have already on the metabolism side of that gulf can answer those questions. They're already very well described. A lot of good work has gone into building cold culture systems where you have stromal layers and then hepatocytes, right? So basically and then folks like Steve Ferguson are doing a lot of work with 3D models, and there's a lot of organotypic culture work. So to me, I think those are definitely the most appropriate sort of models to look at chemicals in a lower throughput fashion. The question I think that we're trying to address is a little bit different, and that is, how do we go about screening 5,000 or 10,000 chemicals and trying to identify the ones with potential liabilities? And then I think I wouldn't necessarily use my cell model, or I wouldn't actually use the models that we're building here to necessarily characterize what's physiologically relevant. I think once we've identified and we can sort of filter that list down to a more manageable number of chemicals, then it would be more something appropriate to basically shift over to the other side of that canyon and use some of the more traditional sort of metabolically accepted approaches.
Great. Well, thank you, everyone, and thank you especially, Steve. We're at time, so we're going to go ahead and close things down. But if you have any other follow-up questions, feel free to reach out to me, and I will pass it along to Steve. I hope all of y'all have a good day, and I hope you'll join us next month.
The leader has disconnected. The conference will be terminated.